the assessment of nematode diversity in orchards. So behind me there are at 20-meter intervals predetermined sampling areas, and we took samples from under the tree and in the middle of the work row. They went to NemLab, and NemLab assesses all the nematodes in that soil sample, including plant parasitic nematodes that we keep an eye on, because there is a relationship between some cover crops and plant parasitic nematodes. So we're aware of that, and Rinus Knutzer at ARC has done some fantastic work on that, industry funded research. The other result that it yields is, because of the different types of nematodes in there, you get a nematode diversity figure. And the NemLab assessment allows you to see how stable that nematode community is, and it's got an enrichment index attached to it as well. So you can see over the last 4 years how the nematode community has stabilized over time and moved into an area, if I can call it that, where it's not over-enriched and it's diverse. And that is what we're aiming at. The planting of indigenous grasses in orchards, I think, is a brilliant idea. There are currently logistic efforts with it, limitations with it, in terms of how do you source seed. It's very easy to source fescue seed. I just go and buy it. I can't get Melina seed easily. I have been messing around with some indigenous plants, and they do quite well in Stellenbosch. Yeah, we have to look. One of the questions that comes up is the influence of cover crop flowers on bee activity during blossom. So Anseli did a preliminary trial on Lorraine in Warmbokkenveld, where we mowed out 4 rows to basically remove cover crop flowers. We left 4 rows open, and we repeated that. And the net result of that is, if you do have flowers in your cover crop, it tends to retain bees in those rows, i.e. your pollination should be more efficient with flowers than without flowers. And the bee activity in those trials showed that very, very clearly. The temptation is to say add carbons in to your orchard. That's true. It's a question of what form you add it in. Putting an organic mulch and compost is going to improve your soil carbon. The thing that's going to put in most carbon are roots and what's associated with those roots. The more diverse those roots, the more diverse the biological community is in your soil, and that is what fixes carbon. Adding carbon in terms of organic material, dry organic material into an orchard, is going to be beneficial, but it's not going to add that much. Remember, a lot of your carbon is labile carbon. It goes away as carbon dioxide. It gets eaten. It oxidizes. It's not going to be there in 5 years' time. We're interested in getting deep carbons and stable carbons into soil. That's a very difficult question to answer. What I don't want is an unbalanced micro community in, for example, under trees. That is going to serve no purpose, because that's where the system can break and you get bacterial diseases, poor growth, etc. What is interesting in that fungal bacterial assessment we did, remember we were monitoring there and there through all these treatments. They influence each other. I would have expected that to be very similar because we're putting compost on every year. They differ between treatments, number one. Number two, that is fairly similar to that, but differs between trials. So there's some interactions there that we're not familiar with.
Hello, so I'm Ansuli, the one Matthew's been asked about the Collembola. So I'm just going to tell you a bit about that. So this is the litter trap we use to collect the Collembola. So basically what we do is we sample a few leaves and then we dry them out and weigh them, and then we put them in here and then we place them in the soil and leave them out for like 2 months. After which I come back and I collect them and then we put them in the funnels facing downward, and then they get extracted into the alcohol. Now the reason we do that is to see what the diversity and the amount of the Collembola is in the soil, as well as what the relation is to the diversity of the cover crops. As you mentioned before, with every treatment there's an increase in cover crop diversity. So I'm going to look at if the cover crop diversity increases the rate of decomposition, and whether the Collembola then also play a factor, whether there's more Collembola, whether that also increases, plays a factor in increasing the decomposition. And then the cool thing about Collembola is they help with the litter breakdown, but they're also good soil health indicators because they don't have a hard exoskeleton. Their bodies are very exposed to the soil environment. And that means that any slight changes in the soil environment means that it directly impacts them. So they are immediately affected and they immediately react to that. So any change in Collembola numbers or diversity can tell us something about the soil as well as the soil health. So that's really cool. And then the other thing is they tend to either eat plant material or they eat bacteria and fungi, which is really cool because they tend to spread the fungi in the soil as well. So say if you have like mycorrhiza in the soil, then they tend to be able to spread the fungi as well. And then the cool thing that I've found is with the increasing diversity in the soil, the number of Collembola has also been increasing. So that tells me something about the cover crop diversity, because the control treatment that we're doing that has no cover crops, the number of Collembola is far less than any of the other treatments. So that's also a really cool thing.